Students, in this class we are going to learn some of the important diseases of mango, and among the diseases which affects the mango, the malformation caused by Fusarium mangiferae, which is very predominant, followed by the anthracnose, which is causing latent infection also caused by Gliosporium gliosporiadis, and then powdery mildew, which affects the tender portion of the mango trees caused by Oidium mangiferae, the stem end rot, as well as the dieback diseases caused by Botrytis theobrome, and grey blight caused by Pestalotia mangiferae, sooty mold caused by Capnodium mangiferae, and the algal disease, that is red rust, caused by Cephalurus mycoides, and all are important. And there is a phanerogamic parasite menace, that is Loranthus, which is a partial stem parasite. Besides, a physiological disorder, that is black tip of mango, is also a problem in mango cultivation. Let us see one by one in the subsequent slides. And mango malformation, which is caused by Fusarium moniliforme, which produces three important symptoms, that is bunchy top phase, vegetative malformation, and thirdly the floral malformation. In the bunchy top phase, in the nursery, bunching of thickened small shoots bearing small rudimentary leaves are the common phenomena, and then shortening and of internodes, and then stunted growth of the plant gives bushy appearance of the top portion. And this is the bushy appearance of the bunchy top phase. And then second phase is the vegetative malformation. The excessive vegetative bunches with limited growth are predominantly noticed, and these are with swollen and shortened internodes forming bunches of crowded leaves with various sizes. The top of the seedlings which show bunchy appearance and which may resemble the witch's broom, and hence it is mentioned as a witch's broom symptom. Mentioned as witch's broom symptom. It is also mentioned as witch's broom symptom. And this is the typical vegetative malformation formed by the pathogen Fusarium moniliforme. And then the thirdly, the floral malformation, the malformed head that dries up in a black mass and persists for a long time. The affected flower panicles become thickened and highly branched, producing up to 3 to 4 times than that of the normal flowers. And these flowers are enlarged, sterile, and they do not bear any fruits. And there are often high numbers of male flowers compared to with the perfect flowers. And this is the malformed inflorescence, that is the floral malformation, which may resemble like a cauliflower's curd like appearance with some necrotic flowers. And the causal organism Fusarium mangiferae, which is known to produce three types of spores, that is Macroconidia, Microconidia, and Chlamydospores. So it is severe during the month of December and January, when the temperature of 10 to 15 degree is prevailing, and the crop of 4 to 8 years are highly susceptible to this infection. And the infestation by the bud mite, that is Aeseria mangiferae, which is act as a predisposing factor. The pores or the wounds caused by this minor, which will helpful for the pathogen to enter and cause malformation symptoms. And to manage this disease, you have to spray NAA at 100 to 200 ppm range during the first week of October, and that will helpful in reducing the floral malformation. Later, 4 sprays of NAA at the rate of 250 ppm at weekly interval from 20th of October onwards. 0.1% of diazinon or monocrotophos or phosphomidon, etc., which will be helpful in reducing the bud mite infestation. And foliar application of carbendazim 0.1% or COC at 0.25% at a later stage will helpful in reducing the spread of the disease. And then second important disease is anthracnose, which produces leaf spot symptom, dieback symptom due to infection and the twig, and then blossom blight, tear stain symptom, alligator skin like symptom appearance, and then rotting of fruit. All are the types of symptoms produced by the anthracnose pathogen Gliosporiatus. Neelam variety is highly susceptible to this infection. This is the typical anthracnose leaf spot produced on the circular or irregular black spots produced on the leaves. Later, the middle of the portion may fall off and leaving the short hole symptom in the affected leaf portions. And the pathogen is also known to produce dieback symptom of inflorescence due to drying from tip downwards, and it also produces irregular or circular black lesions on the fruits, which will later become sunken and causes deterioration of internal pulp. The Neelam, Malgova are highly susceptible to this sort of infection, while the Bangalore is moderately resistant to this infection. And this is the pathogen is also known to produce a phenomena called a latent infection. That is, the pathogen may get attached when the fruit is unripened, but it won't express when it is unripened. But during the ripening process, the outer rind get weakened or thin, and the internal pulp is also turned into sugary nature, which favors the pathogen to establish during the storage condition or during the ripening process. And this is the alligator type of skin effect due to infection by this pathogen, and this is the tear stain symptom. That is due to severe infection of this pathogen. There will be a stain that running from the top to bottom of the fruits, which resembles the tear drops, and hence it is mentioned as tear stain symptom. And this is the pathogen, that is Gliosporiatus, which is known to produce single-celled oval hyaline conidia with oil globules. Wet, humid as well as warm weather which favors the development of this disease, and a temperature of 25 degree and relative humidity of 90 to 97% favors the development of this disease. Warm, humid weather during the during the storage, which helps in development of post-harvest anthracnose disease. And then the conidia that spreads through the wind especially, which is responsible for faster spread. And for managing this disease, prune the trees yearly once and remove the fallen part of the dried infected plant debris, and then wider spacing will inhibit the spread to some extent, and intercropping of mango with other non-host crops reduces the pathogen epidemics. And then spray when the panicles are initiated or appeared, and continue with the recommended interval, and then foliar application of 0.1% carbendazim or 0.2% copper hydroxide or 0.1% copper oxychloride or difenoconazole at 0.03%. All are recommended during the cropping period if the symptoms are observed on the crop. Besides, some of the biocontrol agents which are specific to the pathogen, that is Pseudomonas fluorescens FP7, which may also be applied at 3 weeks interval commencing from October at the rate of 5 grams per liter of water on the flower bunches, reduces the infection ratio. 5 to 7 such sprays are required to reduce the infection. And besides, as I told you, the latent infection is a phenomena in the case of anthracnose infection, and to reduce the latent infection before storage, the fruits should be treated with the hot water at the 50 to 55 degrees Celsius for 15 minutes, or dipping the fruits in 0.1% thiabendazole for 5 minutes will be helpful in reducing the post-harvest infection. And then next important disease is the mango powdery mildew caused by Oidium mangiferae, which is the Oidium type of powdery mildew, and produce white superficial powdery patches on the upper portion of the young leaves as well as stem, petiole, stalk of the panicles and flowers and young fruits. These affected flowers as well as the fruits may drop off prematurely and reducing the crop load considerably, and it might be even prevent the shedding of new fruits. This is the typical powdery growth on the upper surface of the leaf, especially on the tender portions or the young leaves. And this is the powdery growth on inflorescence, which results in the premature dropping of immature fruits. And the causal organum is Iodium mangiferae, which produces ectophytic mycelium and produces conidiophore, short conidiophore with chain of single-celled barrel-shaped conidia in a basipetal arrangement. The primary spread is mainly through the ascospores, which are present in the or the dormant mycelium, which are present in the fallen leaves, and the secondary spread is mainly through windborne conidia. A heavy mist and cool nights during the flowering stage, which favors the development of disease, and the optimum temperature requirement ranged from 15 to 30 degrees Celsius with 70 to 80% of relative humidity. And alternate spraying of wettable sulfur at the rate of 2 grams per liter, or tridimorph, which is not available nowadays, and or bavastin, that is carbendazim, at the rate of 0.1% at 15 days interval is recommended to reduce the disease incidence. The first spray has to be given at the panicle emergence or initiation stage. And then next important disease is dieback, caused by water diplodia theobrome, and later now it is renamed as Lasiodiplodia theobrome, and which is more conspicuous during the October and November months in the mango plantation. And this disease is characterized by drying of twigs as well as the branches from tip downwards, followed by complete defoliation, which gives a scorchy appearance to the tree. And initially it produces darkening of bark, and this darkness advances and extends outward along with the veins of the leaves. These affected leaves turn into dark brown and the margins are rolled upwards, and at this stage the twig and branches are also got affected, which leads to shriveling and severe leaf fall, and this is accompanied with the exudation of yellowish brown gum from the affected barks or crevices on the affected barks. This is the typical dieback symptom due to the infection by Bartonella diplodia or Lasiodiplodia theobrome, and this is the gummy exudation from the affected or completely dried stem portion. And this Lasiodiplodia theobrome is known to produce single-celled hyaline conidia, which are initially single-celled and hyaline. Later, at the time of maturation, that would become bicelled, dark brown in color with numerous striations. The primary spread is mainly through a dormant mycelium present in the infected plant debris, and the secondary spread is through the conidia, especially by the rain splashes as well as the wind current. And high temperature during summer more than 30 degrees Celsius, and prevalence of relative humidity at 80%, which are all favoring the development of this disease. To control this disease, prune out the infected twigs and apply copper oxychloride at the rate of 0.3% on the infected trees. And the cut ends must be, and the affected portions must be pruned, and the cut ends should be protected with the copper oxychloride. Fruit dipping at borax solution, 6% borax solution, keeping it at 43 degrees Celsius, which is also helpful in reducing the incidence on the fruits. And then grape blight or Pustulocyasis leaf spot, which is causing a brown spot at the margins of the leaf, and the tip of the leaf lamina got disturbed, dried irregularly. Initially, these spots are brown in color, later enlarge with dark brown margin and gray center. There are numerous black dots at the gray center, and these black dots which represents the acerulae of the fungi. So the mature green fruits, upon the mature green fruits also, the pathogen is known to produce small brown spots with the gray center, and later turn into larger lesions with numerous number of acerulae. And this is the infection by the pathogen Pustulocyasis mangiferae, and the fungi is known to produce a typical conidia, five-celled conidia. Middle three cells are colored while the top and bottommost cells are hyaline in nature, and the topmost cell is with characteristic appendages which helpful in dissemination of the spores. And the fungi which is present in the stem and multiplies faster during the favorable conditions and spread faster rate through the wind current. Temperature of 20 to 25 degrees Celsius mostly favors the multiplication of this pathogen. And to manage this disease, apply carbendazim at 0.1% after the rains, followed by application of wettable sulfur at 0.2% before flowering, and helpful in reducing the incidence, or even application of Bodo mixture 1% solution, which is also helpful in reducing the incidence level. And then sooty mold. And sooty mold is a formation of black incrustation over the surface of leaf. This fungi produce mycelium which are superficial and dark in color. Even it grows on the flowers and the tender parts, stem, even on the fruits. And they grow or thrive on the sugary substance or secretions produced by the plant hoppers, and establishes form a black incrustation over the surface of the leaf as well as on all tender parts including the fruits, and reduces the photosynthetic ratio of the plant. And during the flowering time it results in reduced fruit set and responsible for fruit fall also. Black coating may also be formed on the surface of the fruits, and this is the typical black incrustation on the surface of the leaf as well as on the surface of fruits. And it is caused by the pathogen Capnodium ramosum, and Capnodium mangiferae, and Trichospermum acerinum. All are predominant, and it forms a black incrustation. It neither establishes the parasitic relationship with the host plant. Since it formed the incrustation, it reduces the photosynthetic value and gained the importance. So the high infestation of the plant hoppers indirectly that is helpful in high incidence of formation of sooty mold. The trees exposed to eastern side have fewer incidences than that of the trees at the center of the orchard. Continuous rainfall washes away these black incrustation formed on the surface of the leaf, and both the insects and the sooty molds are simultaneously controlled by eradication process or removal process. The insects are killed by spraying the carbaryl or phosphomidon at 0.03%, and followed by spraying with the diluted solution of starch or maida, that is at the rate of 5% solution. Upon drying of this maida, the starch comes as a flake along with the encrusted mycelial growth already present on the leaf surface. Spraying of some of the insecticides, which is also helpful in reducing the insect population, and spraying of wettable sulfur reduces the population built up of Capnodium on the surface of the leaves. And then stem end rot, which upon the infected fruit, the pericarp become darkened in color near the pedicel base, and more portions of the fruits which will turn into dark black, and the internal portions are softened and discolored, leading to complete rotting of the end. And it also produces some dieback symptom which studied earlier in Bacterodiplodia theobrome. Similar same pathogen are the Diplodia natalensis, which is also causing this same end rot symptom. The relative humidity of 80%, and the temperature ranges from 25 to 31, which is helpful in development of the disease, besides the rainfall and nutrition deficiency, which is also helpful in development and spreading of the disease. And the plants with balanced fertilization could fight against the establishment of this disease, and the coating on the stem with the fungicidal paints, which will be helpful after the harvest. After the harvest, or packing of the fruits directly into the cellophane bags. The infections after the harvest, packing of fruits in the cellophane bags or polythene bags, which will helpful in reducing the fruit infection. Removal of infected pedicels during the fruit ripening, which also helpful in reducing the spread of the disease. And dipping the fruits in spraying the carbendazim or chlorothalonil at 0.1 and 0.2% respectively in the field before the harvest, which will helpful in reducing the stem end rot incidence. And then red rust, which is a algal disease caused by Cephalourus species. This disease is characterized by formation of initial velvety green round raised pustules on the surface of the leaves and later turn into reddish rusty spots on the leaves as well as on the young twigs. The spots initially are circular and slightly elevated and on the upper surface of the leaf, and it produces, and the pathogen known to produce unbranched filaments which project through the cuticle, and some of these filaments which represents the sterile hair-like structures of the pathogen, while the others which bears the sporangia are the fertile ones. The spores may fall off and leaving the affected portion as a grey spot, and this is the rusty pustules, raised pustules due to the algal pathogen Cephalourus, and after liberation of the spores, these affected area become grey in nature, and the pathogen, as we already learned, that it is an algal pathogen Cephalourus virescens. The algae, which after a period of vegetative growth, it develops on the, it produces the reproductive structures. Certain cells produces the sporangia. There are two types, which was fertile and sterile. Those which bears the sporangia are the sterile fertile sporangiophore, and those without sporangia are fertile sporangiophore. And they are formed on the vegetative filaments of the pathogen and produced just above the surface. The special sporangiophores, which consisting of a thick, rigid septate hairs and swollen at the tip as a vesicle, and this vesicle carries 3 to 6 sporangia on the curved pedicels. When the sporangia are matured and the contents are released into the environment, and these zoospores are orange in color, ovoid, swim actively by means of its flagella. This is the typical sporulating structure. This is the long reddish brown sporangiophore, which produces a vesicle, and upon the vesicle the sporangia are formed with a small structure called sterigmata. And to control this disease, you can apply copper oxychloride at 0.1% or Bordeaux mixture at 1.2% or lime sulfur, which will helpful in reducing the incidence. The bacterial canker or the bacterial spot of the mango. This will also appear on the leaf, but it is very difficult to differentiate the infection of anthracnose with that of the bacterial spot. The anthracnose is irregular circular spot occurs at any point, while the bacteria, which establishes water-soaked symptoms as well as necrotic spots restricted by the veins and veinlets. By the way, we could surrounded by yellow halo. By the way, we could differentiate the anthracnose infection from that of the bacterial spot. The disease that affects all the parts, including leaves, leaf stalks, stem, and fruits. And upon the leaves, it initially produces water-soaked irregular lesions and later turn into black in color, surrounded by chlorati halo. Due to since the spot is limited by the are restricted by the veins and veinlets, that is becoming angular in nature, and which results in the cankerous patches formation, and sometimes the affected leaves may completely dries up. The severe infection leads to defoliation. The pathogen, that is bacteria, which also affect the fruits, There also it produces water-soaked lesions and later turn into dark black and cracking of fruits, accompanied with exudation of bacterial oozes from the cracks. There are a few lesions formed on the fruits and upon the bunches as well as on the twigs also. It produces longitudinal fissures. The variety Peter as well as Alfonso are highly susceptible to this disease. This is the typical necrotic spots. The spots are restricted by the veins and veinlets and they have become angular in nature and typical necrotic spots on the surface of the fruits. And this Xanthomonas axinopodis pathophore mangiferae, it's a gram negative rod shaped bacteria, motile in nature, and it due to presence of monotrichous flagella. Use clean and planting material or grafting material and use certified seedlings to avoid the infection by this bacteria. 2 spraying with the streptocycline 200 to 300 ppm at a 20 days interval reduces the bacterial infection, or dipping of the fruits after harvesting in 2200 ppm of agromycin 100 is also effective against the disease. The mango varieties like Bombay Green, Fazil, Jahangir and Swarna Rekha which are resistant to this disease. And then giant mistletoe, which is a phanerogamic parasite which infects the mango trees. The flowering plant which parasitizes the mango tree at the branches and establishes a strong relationship with the host plant by forming a larger hostoria, which is the absorbing organ through which it absorbs water and nutrients from the host plant. And severely affected plants, mango plants become completely weakened and its productivity got low and ultimately leads to death of trees at the latter stage. And this is the Loranthus which is established on the mango trees. And this is the hostorial point or connection or attachment established by Loranthus on mango tree through which it absorbs the nutrients and water. And the Loranthus, which is scientifically known as Dendrophthia ampullaceus, and there are many other species which affects the mango tree and establishes parasitic relationship. This is the flowers of Loranthus which and the fruits of Loranthus which are all more attractive and hence attracted by the birds, and those birds which eat these colored got attracted by this colorful flower as well as the fruits, and the seeds of this Loranthus are get transmitted to other trees through the fecal matter of those birds which eaten away the colorful fruits produced by Loranthus. And the parasite is responsible for reduced berry formation, and the branches or twigs which are with the parasitic relationship for mistletoe has to be cut off at the point below the infection, and the cut ends should be protected with the Bordeaux paste to avoid further infection by any of the airborne fungal organisms. And the base banding technique which is specifically developed for the mango tree to get rid of this giant mistletoe Loranthus, which includes application of 1% 2,4-D to prevent the xylem translocation, which results in the non-regenerative parasitic mortality, which involves banding at the base of infected branches with the 2,4-D, which helpful in after and this has to be applied after removing the outer skin just below the point of parasitic relationship established by the Loranthus, and followed by 2 consecutive foliar spraying with 1% ethanol or 60% diesel with the second spray with the 60% diesel, which also helpful in reducing the re-emergence. But these treatments are very costly and practically could not be followed. and removal of affected or parasite relationship established branches are predominantly recommended, and the cut end has to be protected with the border placed as we discussed in the previous slide. And then black tip of mango. So among the commercial cultivars, the Deshari is a most common variety cultivar which is prone to this disorder, while Lucknow Safeda, which is very responding very low to this disorder. The infection of the fruit is initiated right at the marble stage with the characteristic yellowing of tissues at the distal end of the fruits. Gradually, the color intensifies, turn into brown, and then finally into black. At this stage, the further growth and development of the fruit is retarded, and formation of a black ring at the tip which extends towards the upper part of the fruit. The black tip disorder is generally been detected in the orchards just located near to the brick kilns, and the smoky gaseous substances released from the brooklins, which consisting of carbon monoxide, which interferes with the boron metabolism and leads to development of this black tip symptoms on the fruits, especially in the distal end or the blossom end. And to manage this symptom, you can apply 1% boron mixture, because just now we learned that the boron deficiency leads to formation of black tip, so you supplement the boron in the form of borax, which will helpful. And then spraying of washing soda 0.5% and caustic soda 0.8% is also helpful in minimizing the black tip disorder. Irrigation in the orchard should be maintained at the proper or regular interval.